Arbor was founded about 5 years ago by Feng Zhang, who's a CRISPR pioneer, and David Walt from Harvard, who is an expert in high throughput automation approaches. And they founded the company on an idea, and that idea was really based on the fact that the original Cas9 CRISPR that was developed or discovered was not the best nuclease discovered, is the first one that worked. And so now we have the opportunity to actually dive into this large amount of metagenomic data and identify truly differentiated and novel nucleases that are better and have more appropriate features for a therapeutic than Cas9 does. And so that was the basis for the formation of Arbor. Since then, we have built a really strong database and platform which allows us to find these novel CRISPR nucleases. And then importantly, once we find them, we need to engineer them to get their levels of activity up to high levels in mammalian cells. So we've got this really important platform that allows us to do that, combining that with a portfolio view of the space. So we have a portfolio of these editors, and that allows us to look at the disease pathology first, and then tailor the appropriate editor to that disease pathology. So an example would be if we looked at something like a triplet repeat disease, where there's a massive expansion of a triplet or a three-part codon within the DNA in some patients like Huntington's disease, for example. And if that's what we know we need to do, we can then tailor the therapy to that disease. So we can pick the right nuclease that will actually bind to that area. So it has the right, what we call a PAM, or a binding motif. And we pick the nuclease that has the right cut properties, so it can actually excise and remove the DNA that's needed in that case. So that's how we think about tailoring, which is important to have this portfolio approach so we can actually hit any disease anywhere in the DNA where we need to.